Welcome to the layman's presentation of my PhD defense. I am Michel Bengtson, and the title of my dissertation is Development of DNA Diagnostics of Neglected Tropical Diseases in Resource-Limited Settings. My PhD research was funded by the TU Delft Global Initiative, which is a booster platform of science and technology for global development. The goal of our research is to develop concrete solutions for worldwide challenges and to make impact. My dissertation is composed of 6 chapters. In chapter 1, I provide an introduction to the topic. In chapter 2, we provide a review of point-of-care diagnosis of neglected tropical diseases. In chapter 3, we look at context-driven design for the end user. And in chapter 4, we look at the scientific methodology that is based on CRISPR-Cas9. In chapter 5, we look at the inhibition of CRISPR-Cas9, and in chapter 6, I provide a general perspective. These are the Sustainable Development Goals, and this research is geared towards Goal 3.3, which aims to end epidemics of AIDS, TB, malaria, neglected tropical diseases, and other infectious diseases. When looking at the global map of neglected tropical diseases, we see that millions of people require interventions against neglected tropical diseases. And we also see a trend of prevalence in the global south. Chapter 1 takes a closer look at diagnosing infectious diseases. Effective treatment and prevention of these diseases requires accurate diagnostics. However, current diagnostics are expensive, time-consuming, and they require equipment and resources that are simply not available in resource-limited settings where they are needed the most. The WHO have developed various guidelines, such as the Assured criteria, which aim to guide the development of new diagnostic tests. When looking at different diagnostic settings, point-of-care testing aims to decentralize diagnostics by taking the test out of the laboratory and bringing it closer to the people that need them the most. In chapter 2, we see the healthcare paradox for neglected tropical diseases in endemic regions. Basically, at the base of the pyramid, where there is the highest frequency of need, there is the lowest availability of resources. We also take a closer look at all of the NTDs, and we aim to find point-of-care implementation needs that could be applied to more than one disease. Throughout this research, we look at leishmaniasis as a case study. There are more than 20 different species that cause this disease, this group of diseases. There are 3 forms of the disease. Visceral leishmaniasis affects the visceral organs, and it is fatal if left untreated. Cutaneous leishmaniasis, although not fatal, is still extremely debilitating and has a very high social stigma. Collectively, there are approximately 1.2 million reported cases per year. Although treatment is available, there is a lack of effective diagnostics for this group of diseases. Taking a closer look at resource-limited settings, people travel 70 kilometers to the nearest healthcare center. In chapter 3, we provide a context analysis of our field research in East Africa, where we traveled in Kenya and Uganda. We chose to go to East Africa because current diagnostic tests perform quite well in India, for example, but they don't perform as well in East Africa. I wanted to share some of the pictures of the community life in Baringo County in Kenya. On the top left, you can see that people live in very close proximity to the breeding sites where the vectors that spread this disease live. You can also see that these settings are extremely resource limited. In chapter 3, you see our context analysis, and you see how we used the context to give us requirements, design requirements for the new point of care test, and also to see where the new point of care test would fit into the context. And the take-home message here is that design thinking can facilitate bridging the gap between technology and the healthcare system where the technology will be used. At the heart of this research, we have developed a DNA-based detection scheme. DNA detection for diagnostics has advantages over immunological diagnostics, for example. By directly detecting the DNA of the pathogen itself, this becomes independent of the person's immune system or the person's health status. When developing a point of care diagnostic test, we have to think about the sample, and what we aim for is a non-invasive sample extraction. And here you can see a pinprick of blood or a urine sample, which can be obtained from a patient in a non-invasive way. And what we've developed is a paper-based membrane wherein we are able to trap DNA as well as proteins and other cellular components. And with only one step, by adding a buffer or a liquid, we are able to release the DNA from the membrane while still trapping the proteins and the other cellular components. So taking a closer look at the DNA, we chose to target kDNA, kinetoplast DNA, wherein there are multiple copies per parasite. For this specific target that we chose, there are approximately 10,000 copies per parasite. So this allows us to potentially be able to detect even a single parasite because there are so many copies of this gene. And to couple this DNA detection to the rest of our diagnostic scheme, we use CRISPR. So CRISPR has received a lot of attention recently, especially because of the Nobel Prize for Chemistry in 2020. And basically it's a protein system and it functions as a scanner. So if you think of the sequence of the parasite being a unique barcode, CRISPR is able to scan all of the DNA sequence to find a match. The next step in our workflow is to couple this detection to a readout that is visible to the naked eye, because we want an equipment-free diagnostic test. And we were able to get some very promising results. You can see a very clear readout. There's a clear difference between a sample with or without the parasitic DNA. And although there are limitations, for example, we only tested one patient sample, further validation is required, but the results are very promising. So looking back at the context, in its current state, the technology would need to fit in a hospital or a treatment center where there are some resources. Although our detection scheme works at room temperature, you don't need any external equipment. It does require some time. It requires an overnight incubation. So at the moment, the assay is not optimized in order to be used, for example, at a local homestead, but it could still be very valuable in a hospital or a treatment center. And I'd like to share some of my reflections with you. At the beginning of this PhD, I was at the peak of inflated expectations. And about midway through the research, I was in the trough of disillusionment. And now at the end, I am at the slope of enlightenment. And I would like to end by thanking all the people that have been involved in this research, to my supervisor, colleagues, collaborators, the students who have been involved, and especially stakeholders and our local stakeholders. I thank you so much for your contribution to this research.